Hi everyone, my name's Nat, and I am the head of community at an organization called Exceptional Individuals. We support neurodivergent individuals and organizations. And for the last few weeks, we've been getting really nerdy and diving in deep to the origins of neurodivergence. When we talk about neurodivergence, we're talking about dyslexia, dyspraxia, autism, ADHD, but we're normally talking about how we support people who are already diagnosed or resonate. But we've been trying to really try and get under the hood and figure out what really is neurodivergence. Where does it stem from? How does it develop? Where is the root cause? Now, you might say whether or not that's important or not, and I think that's a personal choice. And honestly, we don't have an exact answer, but this is a really great way of opening up the science side of things. Now, a bit about my background. I am neurodivergent, and I'm also a neuroscience mastery. I don't know what I'd call that, just a student. So I'm currently doing my master's in neuroscience, plus I support neurodiverse people every single day, and I'm an educator. So fingers crossed, I'm taking a very complex subject today and trying to make it as digestible and easy for everyone, while also still hitting home the key points. But if anyone is confused at any point, just let me know and I'll try and reword it so we're all following at the same time. So as you can see by the screen, normally our bread and butter is dyslexia, dyspraxia, autism, and ADHD. That's just the areas that we specialize in. But we are coming at it from a neuroscience perspective today. And while we're not really going to be focusing on any of these as such, the main reason we're exploring neurocells and glial cells is to understand at what point does a kind of disalignment, damage, how it develops, translate into what will later be how we process the world. Once again, it's we're still researching. There isn't a definitive answer, but we do know there is a link for sure. Now, here's our lovely team, also neurodivergent. Do check us out on our website, exceptionalindividuals.com. And we also record webinars every single week. So the last one we did, which is now live, is on the brain anatomy. We got the brain, we sliced and diced it, and we labeled it with all different names. So if you want a nice entry-level way of learning your brain basics, do subscribe to our channel. So let's start off with neurons and glia. Oh, I don't know where all of your knowledge is at the moment when it comes to neuroscience, so I'll try my best to keep it at a level which is accessible for everyone. But essentially, in our brain is the neuron region, and in our brains we have lots of little cells, and those cells have other cells, and we are going to be talking about the cells in our brain that essentially does everything for us, does the thinking, the processing, sending the messages. And by understanding these two better, maybe, just maybe, we can understand neurodivergence a little bit better. So that's the goal, at least. So hopefully we're going to be covering how do we even talk about this so-called nervous system, normally abbreviated to NS. The nervous system is one of the oldest parts of the body in terms of evolutionary speaking. It counts for the brain and the cord that kind of goes down your body. That's the nervous system. It sends all the information to where it needs to go. We've also got subdivisions, so we can split it up even further. So when we talk about it, it actually makes sense. We've got different cell types in the nervous system, such as neurons and glia, and we've got different neurons and different glia cells. So breaking it up even more. We've also got something called intracellular components. Now that sounds a bit confusing, so I broke it down further. That just means structures and substances that exist within the cell. So maybe that's the chemicals that kind of send the messages. Now let's kickstart with our first question. These two fellas are the fathers of neuroscience. Bless them. Have you ever heard of them? Do not worry if you haven't, just want to get a feel of the room. These two people both won the 1906 Nobel Prize in medicine. Now, interestingly, they were like arch enemies, both doing their own research, but ultimately their research was really useful and interesting. Oh, no one's heard of them. They're fascinating chaps. So here is one of the drawings that one of them did. And you can see them starting to draw what the neurons in our brain are. Remember, neurons are just the connector cables that help us, our brain, communicate. And they were doing pioneering research by slicing the brain up, adding bits of dye, and allowing us to actually see what the brain looks like for the first time, and kind of drawing it in a way which everyone could see how the brain connects. So in these drawings here, basically these long squiggly lines are neurons. Then the long parts are the axons. We've got the dendrites, which are the bits that kind of go off, which accept the signal. They're beautiful bits of art in their own right, but a massive step. So what we're talking about today, we have come a long way from when these two first got their Nobel Prize. Now, on this screen, this is a photograph, obviously super magnified, and it shows the massive components in the brain, all the billions and billions of tiny little cells. And if you're able to, I want you to put a little pin on your phone where the nucleus is. Okay, I can see we've got one person already. Nice, nice. Now you can kind of see, okay, so if you see the dots, the dark ones, yes, they are like the nucleus, and there's lots of them around here. um, oh, and you can kind of see, and that's essentially the hub. That's where like the information is stored, like the essentially the brain within the brain. On the next one, do you know any of these nervous system cells? Obviously, I've been speaking about neurons and glia, but I'm just curious to know which one are you familiar with? Yeah, I'm not surprised. Most people are most familiar with neurons. They are the ones that most people have heard about. But glia cells, no one's ever heard of glia cells, but they are so, so important. They completely outnumber all our neurons. We might have millions of neurons, but we have billions and billions of glia. They are essentially, or what used to be considered, the scaffolding of the brain. So you got a neuron and you got another neuron there touching, they're going to need a bit of support. So the glia cells will help support them. But they also have their own function, and they also play a vital role, which is still very new and still a developing area. So this is why you can't really speak about one without speaking of the other. In this, we put neurons and glia cells in the nervous system transmit signals, provide support, and regulate body functions and behavior. So pretty damn important when it comes to maintaining a healthy body and deciding how our brain is going to operate. And you might have heard, when we talk about neurodevelopmental conditions, you might have also heard of things such as acquired dyslexia or acquired autism. And it's not actually like autism or dyslexia, but they show the same characteristics. So it can show you that even if you were to damage some of these things later in life, you could still have the same characteristics. So it works both ways, either from when you're born or from later in life. Now on here, we'll see a long old neuron, and I want you to give a guess in terms of how fast you think a neuron can travel. If any of you are a keen eye, I actually do hide the answer somewhere, but either way, you'll see here. So these are both different types of neuron, right? And you've got the nucleus there. We've got the dendrites, we've got the axon, and as you can see, they're sending electrical signals down the cable. Now, this one on the right-hand side is doing the exact same thing. But if you notice that electricity is going a lot faster, and that is because of these little kind of jackets that are around it called myelin sheath. And essentially, they act as like a cable for like insulation. So if without your jacket, you're pretty slow. With the jacket, really, really fast. So a lot of you, or all of you, have done 300 miles per hour. And, that's nearly that. They can go nearly 300 miles per hour, but they can also go 1 mile per hour. it varies. It depends on a number of factors. It depends on how strong the connection is, how much signal is being activated, the chemicals which initially start the chemical reaction for the electricity. A lot of it depends on the myelin sheath. How well is it insulated? So depending on how the neuron is structured, depends a lot on how fast it is. So not all neurons are created equally. It doesn't mean that they should all have the insulation. Sometimes slower neurons can be a benefit, but more often than not, the faster it activates, the quicker the reaction happens. If I'm in my brain here and I want my toe to move, if I went 1 mile per hour, I'm going to have such a delay and it's going to make walking a real pain. I need that main one to be super fast in order to be very responsive and active. Now, it's interesting when you look at, say, neurodevelopmental conditions such as dyspraxia, which often relates to motor coordination and being a bit slower to process, you can kind of see how these things potentially might link up. Now, why do neurons even bother passing information? And it's interesting because a neuron and a neuron, right? You think they kind of like holding hands to pass information. They actually don't completely touch. There's actually a bit of gap. So the line sends like electrical signal, and that little gap sends a chemical across. So they use a combination of electrical signals and chemical signals. Electrical is with inside the neuron, and chemical is that tiny gap in between. So neurons use electrical signal within themselves and chemical signals between each other to transmit information. And please do stop me if you want me to kind of go over anything again. I appreciate this is a brand new subject for a lot of people, and hopefully maybe a refresher for some of you. Now, on this one, there's 3 little images. So what is a cell in your brain that talks to other cells? Now, neurons are specialized based on particular functions. Okay, I can see we've got a mixture here. Yes, Brittany, it's all of them. So most of us will recognize the middle one as our kind of go-to neuron, but there's many different types depending on the function, depending on the location, but you'll see that they all share some things in common. They all have a nucleus, they all have an axon, they also have dendrites, they all have synapses. It's just kind of the order, the location, the strength, the number of them. So do not be fooled. Though we're normally always showing the middle one, they can be very different. So different types of human neurons vary in appearance, size, expression patterns, signaling molecules based on location and function. Just like people, we all look different and have different functions, I suppose. Next one is understanding a bit more about how we all look different. So we've got the relay neuron. Relay, when someone passes a baton, another person runs, passes the baton, runs along. They essentially send the information. The chemicals get pumped to the dendrites, these little spiny bits. Then it goes through the cell body. That's the main part with the nuclei. Then it goes through the axon, and then it gets to the synapses, which essentially pumps the chemicals over to the next dendrite. And on we go again. And that goes all through the body. Then we've got a motor neuron. And motor neuron, as it would suggest, this is, kind of helping us move, kind of telling us we should go. And if you notice, whoa, what do they have here? They've got these lovely little jackets on, the myelin sheath. And that is because these neurons, because it's about coordination, they have to be faster. So they are built in a way which means they are super fast and makes sense. But obviously, the faster things are, the more energy they might consume. So it's not like we'd want this for everything. Now, you see here it says the nodes of Ranviera. Well, sorry about my pronunciation. But essentially, we need little gaps in order for the Glial cells, the scaffolding, to kind of hold on and to create little gaps where the signal can pass through. So the gaps are perfectly normal. And then we've also got our sensory neuron. And again, very similar, but things are done in different directions for different purposes. But yeah, they're kind of cute little things. I wish I had my toy today. I got a little toy neuron. It's adorable. All right, so now we hopefully understand neurons, which are, your ABCs of neuroscience. You got a brain, you got neurons inside. Now let's do their Robin to their Batman, the Glia cells, the partner in crime. Now these guys do not get as much love and attention as they honestly deserve, but without these, a whole brain would probably just kind of collapse like a pile of dominoes. They are very much needed, but the amount of them is a lot. I think one of the reasons we know less about them is that they're a lot smaller. And as such, you had to wait for technology to develop further before you could get to know them a little bit more intimate. Now, as you can see here, this picture is a bit chaotic, but you've got your neuron. You can see the myelin sheath there. Oh, yes, April remembers it. And you can see the neuron. And as you can see, you see these blue things there. These are, as I mentioned, think of it like scaffolding. They're essentially holding all the neurons together. And these are very much needed. But do remember, while I will refer to them as scaffolding, they're not just, they actually do have functions, and research is currently going on that says they're a lot more important than what we give them credit for. But as I said, they aren't solo. Think of Glial Cells as the neuron wingmen. Don't get me wrong, neurons are the brains of the operation, but without their wingman, without their sidekick, who would they even be? So some of the other cells we're going to be looking at is Ependymal cells, Astrocytes, Microglia, and Oligodendrocytes. All right, I'll leave you all to read them yourself, okay? Again, why do we have a dyslexic teaching science? I don't know, but here we are. So essentially, just like neurons, we've got lots of different types depending on the function. We also have lots of different Glial cells depending on the function. So not all images you see are going to look the same. Let's have a little bit of a deeper dive into this. Oh, nice. We've got a comment. Glial cells have started getting a lot more attention since the discovery of the glial system. Yes, I'm sorry about pronunciation, but yes, you are right. Now, let's start off with the Ependymal cells. And these cells are like the wallpaper within the brain's rooms, the ventricles and the spinal cords, the pipes in a central system. Essentially, these are everywhere. They kind of like the lining, and they kind of like stick to the outside, create a good amount of structure. I can't give you too much information about these. This is just a real like entry level, they exist. So these cells exist. Then we've got the astrocytes, and you see these guys, look how happy they are. And they say there's over 5 times more of these than neurons. 5 times. And considering how many neurons there are in the brain, which is like billions, this is a lot of neurons. I think maybe trillions. There are a lot of astrocytes, and they're all smiling. They're all happy. So these fellas, they send nutrients from the blood to the neurons. Our neurons, our brains, they don't just run for free. They do require energy. So these little glial cells will send the energy and allow our brain to keep doing that amazing processing. They do the maintenance of extracellular ions. So they're also cleanup crew, and they also help fix things, make sure everything's working hunky-dory. They also do a bit of tissue repair. Our brain is always getting damaged, and we need someone to help it out. So while the neurons might be the stars of the show, these Glial cells are like the handymen or women. They are the ones who are keeping this ship afloat. They might not be the captain, but without them, we have no boat. And they also regulate the synaptic activity. So the synapse is, where one end sends the information to the next dendrite. They kind of keep that working. We also got microglia, and this one a lot of people have heard about. Here's a kind of an image you can see. You can see the neurons, but then the purple bits are all kind of keeping it all together, keeping the structure. They also live in the brain. They're kind of the cleanup crew. When parts of your brain cells die, if no one cleaned up after it, we just have loads of dead neurons floating in the brain and other bits and bobs, or maybe neurotransmitters, which are the chemicals. These guys will help clean it up. They get other cells to areas of damage. So if we're talking about these cells being kind of like the janitors, think of the microglia as like the head janitor. They might not necessarily do the job themselves, but they will get other cells in order to clean up the mess from the neurons. So really important. They're kind of like head sidekick. They also support tissue repair. Really important. And remember, if we're putting all of this under the lens of neurodiversity, if some of these aren't working, you can probably see that someday, somewhere down the line, is going to have an impact on us as human beings and how we operate our physical world. We also got oligodendrites, and these generate myelin sheath. Remember, we talked about this myelin sheath, the nice jackets which make things nice and fast. They generate the myelin sheath to support and insulate neuron axons. So here you can see our neuron, and here we've got the myelin sheath. And normally in images, we only see these little puffy jackets. You don't normally see the bits that are holding it together. Again, imagine that, like you're the person doing all the hard work, but you don't get any of the glory. But they are attached. They increase speed of neuron signaling. And remember, we see a lot of these in the motor neurons, the parts that help us move. Maybe, again, related to parts of neurodiversity, particularly because coordination isn't always the strength of some. We've got provide metabolic support. What do we mean by that? Essentially deliver nutrients, removing waste products. So these guys, they're like the scaffolding. They're also like the waste management. They're kind of the grunts, to be honest. These would not be the head honcho. They would take a secondary role, but equally just as important. So you need all of these Glial cells in order to support our neurons, in order to support our body functions, and to allow us to live a full, healthy, and happy life. Whew. All right. So now dysfunctions in these cells can lead to neurodevelopmental conditions. This is the money part. So when we're asking ourselves, where does autism, where does dyspraxia, ADHD, dyscalculia, Tourette's, OCD, where do these things come from? Well, normally it comes from our brain having some sort of dysfunction or deficit in one part. It might be from when our cells are originally created or from potential damage later in our life. But if some things aren't able to communicate as effectively, they are going to have a knock-on effect to other elements of our life. Have we worked out yet? Like, for instance, exactly what cell and glial cell damage equals what reaction to a human in like real life scenarios? No. I couldn't tell you, you damaged your microglia in this region, now you're going to have ADHD. It doesn't quite work like that, but at least we know something. It's an emerging field, and we know something. So let's have a little look at the cell structure and function. So here is a picture of, remember we talked about the nucleus inside the neuron? This is inside that. So in whatever area we look at, we can get deeper and deeper and deeper. So you might look at your brain. Brain, all right, head, there's no dents in it. We're all good. Then you might look at the brain. Okay, the brain looks fine. So far, no developmental conditions or disorders. Let's go deeper to the neurons. Neurons look fine. I don't see what's the matter. Let's look at the glial cells. Let's look at the nucleus. So you can see why it can be very, very difficult in order to know if someone has a neurodevelopmental disorder just from, say, microscopes or scans or tests, because what we're looking at is so, so small. This is why we normally have to use psychiatry and just ask about traits and do a bit more of a high level, you are very likely to have XYZ, rather than saying definitively you have this, because we do not yet have the information to know what that is. So now let's talk a little bit more about axons, and we're going back to the neurons now. And what I wanted to do is dive in a bit deeper. So far, I talked about neurons and I've talked about glial cells, but we're going to zoom in even further to see what other things can go wrong in our body in order to equal someone like myself. Now, whether or not you say wrong or different or alternative is up to you. I would say being neurodiverse is a positive, But ultimately, in order for this brain to develop, some things had to not talk correctly to each other. So axons. Every brain neuron has a long extension from its core. So remember, you've got your kind of cell body here, the brain. You've got your dendrites. They're kind of, think of these like arms. These are the things which accept the signal. The axon, which is essentially the long line. It's your long part of the body. And that is needed in order to get to A to B. And then lastly, the synapses. So when we're talking about axons, this is essentially just the highway, the motorway, the thing that takes you to A to B. Now on here, I want you to have a guess on how long you think the longest axon is in the human body. So remember, let's say this is part of your brain talking to another part of the brain. Do you think it's 1 nanometer? Couldn't even tell you how big a nanometer is. It's like really, really small. Or 1 meter, which is about, if you put one hand to like a nipple and put your hand out, that's about a meter. Okay, interesting. Now, in fact, the longest axon in your body is 1 meter. That's long. That is literally like that long. Because if you know, you've got your central nervous system, which is your brain and your spinal cord. So from the end of the spinal cord, all the way down to your big toe, that is where the longest one is. And it makes sense because, again, if you want to move your big toe, you do not want any delays, and it has a long distance to go. So if it's having to go from, say, one neuron to the next neuron, a relay neuron, that is going to take a long time, because every time you pass from one to the other, you have which big toe? And it's a good point. I don't know, maybe just one big toe. I imagine they kind of sprint off. But you want it to get there really quick. But most things, you'll have like one, then another, then another. Think of like Lego blocks. But for the main one, from top to bottom, think of it just as a one gigantic Lego brick. So the sizes can vary dramatically. It kind of sounds like you've got a giant tapeworm in your body, but honestly, it's a good thing. It also means that, when people have like spinal damage or nerve damage in the spine, it's like really bad. It's because this is one nerve system you really do not want to damage because it is so essential. Now, let's have a little look at the dendrites. And remember, the dendrites are the hands. These sprout off from the cell body, and these are the things which kind of accept the signals. So the dendrites are the talky bits. The bits that kind of accept the information. And as you can see, they're kind of like tree roots. They sprout off into many directions. And they kind of go all over. All right, so yeah, the dendrites are the bits that send the information. I kind of see it like a kite. You got your main kite body. Anyway, so these bits are also very important. Now, as you can see, there are so many places in our brain cells which could go wrong. Could it be the hands? Could it be the feet? Could it be the body? Could it be the brain in the brain? It could go wrong in many, many places. So when I say neuron input, it's what ones are able to take in the most information. Have we got the cortical pyramidal neuron or the cerebellar pumpkin neuron? A or B? Let's just go with A or B. And yes, as most of you have correctly, well, all of you have correctly said, it is the second one. And how do you know that? Well, if you look at the sheer amount of offshoots, I think every single one can take different information. So the more hands you got, the more things you can carry and the more information you can pass through. So you've got some which kind of like a big tree, and then they all kind of get funneled down one route. And then you have other ones which kind of are like a lot less, and different ones require different amounts and can take more. Let's say but that being said, if you have loads of branching, but they're really weak branches, they might not be that effective. So sometimes you might actually want to have less branches, but stronger branches. So do not always think more is better, but generically having a bit more normally means you can require more. Hopefully this is making sense. Now we're going to look at some spines. So we are getting even deeper. So we've got a microscope and we have just kind of changed the settings to be even more magnified. At the end of the dendritic spikes, we have little spines. No one ever shows you these images, but trust me, they are important. So think of it like, on your skin, you have tiny, tiny hairs. You can't see them, but they are important because they help you with sensation and they let you know if you've got goosebumps and, they can keep you cool. These little spikes are just as important. So the little bumps on the dendrites are part of the synapses. So if you think about it, if we are saying that the dendrites are like the arms of the neurons, then the spines are like the fingers. So if we only had arms, they're pretty useless. But with fingers, now we can actually do something. Now we can actually pick things up and interact with the world. Just like with dendrites, with the spines, they can actually start to take in the information and start making our body do its magic. Another way you could look at this is kind of like a cactus. So on here, these are all different types of dendritic spines. So when I say synapses, this is just the part that kind of sends the signal at the very end. So which one of these little things, we've got a little squiggly line, we've got a long thin one, just a thin one, little stub, a mushroom or a branched one, do you think is going to be able to send the strongest signals? I would kind of see this as having one having like a crooked finger, one having not long fingers, one having a bit of the stubby thumb, another one having an extra finger, more the merrier. So actually, really interesting, you are all correct. So the mushroom one is the most effective, and simply the bigger mass, there's a bigger amount of it in order to take. So imagine you having an extra large hand or a big old baseball glove, you are going to be able to catch more things. Or if you have more fingers, I'm assuming maybe you can catch more. I don't know. Maybe, there's so much fingers, enough fingers. But as you can see, this works. So a dendrite spine is a small protrusion on dendrites which form the postsynaptic side of a synapse, receiving input from another neuron. So when we say postsynaptic, remember post is just after. So after the synapse is the dendrite, after the dendrite is the synapse, after the synapse is the dendrite. So we're just talking about head and tail, essentially.
I have a question.
Yeah, go for it.
It's going back a few slides, but it was the one that had numbers, and I didn't see 6 through 8. Was there a reason for that?
6 through 8.
Yeah.
Could you explain a bit further? I'm not sure.
It had 2 4s on the left and right side of it. It was like a diagram.
This?
That one.
Oh, yeah. So the numbers are. This was taken from an image which will tell you what each of them mean. So number 2 is the nuclei. It's irrelevant for this, to be honest. It was just part of the image.
Okay. Thank you.
But good question anyway. And Anna says, do people with sensory issues have more dendrites on some of their neurons? Yes. I mean, in terms of the main thing, it's a multiple of factors. But yes, the more dendrites you have, the more information you're able to take in. But let's say you are taking more than you're able to process, you might be hypersensitive. Or if things aren't functioning as they should be, you could be less sensitive. It could be that when the one dendrite that one synapse is sending the information to the next dendrite, maybe they're sending too many chemicals, not enough chemicals, or maybe the chemicals get lost. So in terms of why we have sensory issues, honestly, it can be a number of things because the different parts can go wrong. It's also why, though, that when we take medication, it's different for everyone because doctors will not know what's wrong. They'll know that there's sensory issues, right? They won't know why there's sensory issues. So they'll try medication that will make the signals go faster. If that doesn't work, they'll try medication which kind of blocks it to kind of stop information kind of going so rapidly. Other times, they'll do medication which means that when the chemicals are getting recycled, they get recycled better or slower or faster. So it's kind of a bit of a trial and error. And what doctors normally do is they pick the medication which is most commonly beneficial. But if it doesn't work, that's why they'll try something else because they normally work in slightly different ways. It's not all about branding. There's actually method to the madness. Okay, we're going back to glial cells. So we just did neuron cells where we went even deeper and we looked at what's all the little extra fingers in these neurons, such as the spines. Now let's do the exact same things to the glia cells. So here's a lovely glia cell, and again, very much scaffolding-y like, just a bit of glue holding things together, or so we think. Glia cells change shape when they activate or reactivate. So these, they can change. They're like Morphin Mighty Power Rangers. They can become round. They release proteins to attract other glia cells to injuries. Do you remember how I told you how these cells are like the janitors, but some of them are like the head janitors? They'll kind of clear up all the messes. And essentially, they were like, "Oi, we need more people over here to come fix it." But rather than shouting, what they'll actually do is sending proteins, delicious, lovely, beautifully scented proteins, which kind of gets more glia cells, more backup in order to help clear the mess, which maybe the brain has created through like death, pruning, whatever. And when I say death, I don't mean of the person, but of the individual cells. They can also destroy any debris, and including synapses. So debris is just leftover synapses, these like chemicals that we talked about. Sometimes there's too much, sometimes things don't work out, and they can't just stay in the brain. They need to be recycled and reused. Okay, we've got what is the stimulus for change in their shapes and how enduring are the changes? Oh, good. That is a good question. Well, I'm not sure I can answer that right now. I think it will take me a little bit. Maybe I can answer that later for you if that's okay. But good question. The question was, what is the stimulus for changes in their shape and how enduring are the changes? So essentially, why do they change? What provokes it? And how long do they change? Honestly, I'm not entirely sure, but I would love to find out. So honestly, great question. So out of these microglia ones, which one do you think is the most reactive? And we're not talking like Incredible Hulk reactive or like nuclear. These are just ones which go from essentially chilling in the brain to, all right, Avengers assemble. We actually have to do work now. So you've got your janitor sitting, playing on Instagram, just swiping up, to the janitor actually cleaning up and doing their job. Okay, we've got the ramified aneuploid. It is, interesting. It is the one that is the brightest green. So yes, so you can see here that this one is the most reactive. Remember how I told you they go circular? As you can see, at the moment, they're all kind of branched out and then they kind of like close in on themselves. And this is how we can tell if your microglia is actually working. Because when we do scans and we put different dyes in the brain, you can see which ones are active, and which ones are reactive, and which ones aren't doing anything at all. So this is how we can tell, is the brain doing what it should be? Now, in terms of structure, now we're going to look at the cell substructure. So how do we break it apart even more? This is like a nuclei, but you can see inside there's a lot of information. And if we're looking here, remember actually, as one of you rightly said earlier, what did all those numbers mean? Now I can tell you what they mean. So those numbers were on each one of all of these, right? And all of these have their individual name. So this is nuclei inside the neuron. And I want you to put a pin where the cell stores its genetic information. I mean, you can probably kind of guess genetic information if you kind of work out like the letters. But I'm not going to read all of these because honestly, I'll probably butcher them. But you can see our brain of the brain has a brain also. And when we talk about DNA, everyone loves a bit of DNA. No one really knows what it is. We only see those images of like the wires intercrossing. The DNA is inside the nuclei, and this holds all the information about who we are. So remember, in all our like brain cells, they have DNA in every single one. And because these are so small, we leave DNA everywhere we go. So yep, absolutely spot on. So inside a cell body, we have as followed. The nucleus. This is the brain. This stores all the information. So like the hard drive. Actually, I wouldn't say a brain, like a hard drive is more accurate. It has the information and it can be retrieved when needed. And when needed, then it can pass that information along, like the axon and the dendrites and, et cetera, et cetera. So the nuclei, very much like the hard drive. Then the endoplasmic reticulum is the protein produced for delivery. So that is here. I remember proteins are really important. We need these to get everything to work together. Proteins are kind of like the keys. Then we've got the Golgi apparatus. That's just another sorting center. Think like Amazon needed more room, so they kind of expanded. Then we've got the mitochondria, the energy generator. All good buildings need an energy generator because nothing works for free. Everything requires some level of energy. Then we have the lysosome. This degrades protein when faulty. So where is that? Here, think of this like the recycling center. Again, nothing is used, nothing is completely trashed. They're not like us, which I'd like, we're terrible. But our body will use everything. And then the cell membrane, which is like the outer layer, which keeps everything nice and safe. So this is, in basic, what is going on under the hood. In every little cell we have, we've got the talky bits, we've got the bits that send the information, we've got the bits that send more information, and we have inside its own mini factory. There is a recycling center, there is a storage center, there is the main brain, there is everything in every single neuron. And remember, we've got like billions of these in our brain. So there is so much going on in why the human brain is so, so complex. Whew, all right, now for a little bit of true or false. Unique features of a neuron. How well do you know your neurons now? So neurons require a lot of energy. True or false? Neurons transport cargo over long distances. Neurons are resilient to stress. Oh, Anna says, the other two you have not mentioned on the diagram look the same. Actually, one sec. Which ones? Oh, those things. Oh, those. And oh, no, they're not the same. See, one is pointing to the little black dot, and the other one is to the plasm, like the kind of liquidy goo which is stored within them. All right, nice. Okay, let's see what you all said. So we've got, let's see how you all did. Oh, I see, unanimous on both of them. So the first one, neurons require a lot of energy. Yeah, they do. Nothing in the brain works for free. Everything requires energy, proton, it's all needed. And, some people have more energy than others. Neurons transport cargo information over long distances, absolutely. And if any of you have ever ordered something from, say, China, if you're in the UK, it normally gets damaged on the way. It's a long journey. Things get lost. Things go missing. So the longer somewhere has to go to transport the information, the higher likelihood it's not going to get there in one piece or might change along the way. So when we talked about the central nervous system and the kind of longest axon in our body, think of that like an aeroplane, a lot safer and a lot quicker. But yet some neurons might take an old tugboat across the ocean. Going to take a lot longer and probably isn't going to be as good with the final package. And the last one, neurons are resilient to stress. Unfortunately, no. Stress is bad, yeah. Stress is genuinely one of the main reasons for schizophrenia. It can also affect other neurodevelopmental conditions. When we talk about neurodivergence being lifelong, but can fluctuate throughout your life, stress is one of the key factors which can kind of activate the fluctuation. So, yeah. Put another way, the unique features of a neuron include specialized structures such as dendrites and axons, as well as the ability to generate and propagate electrical signals known as action potential. I don't think that actually helped, actually. But as you can see, our brains are complicated. They're not as sturdy as we think. Now, there's an interesting question. Can we make new neurons? Well, yes, we can. And that's a new neuroplasticity and neurogenesis. That's like kind of a relatively new idea that isn't completely proven. But what we do know is it's not easy. So most of the neurons you are born with are probably most of the neurons you're going to have, even though they are dying all the time. But this is, still developing. Oh, yes, Brittany said that's why it's neuroplasticity. Neurons die off. Absolutely. Absolutely right. So some takeaways from today, because we have covered a lot, and it's obviously it's always a challenge having such a diverse audience with depending where we're all levels. But hopefully it was easy enough for those who are new to the subject and still fascinating enough for those who felt that they had a good grip on it. So some takeaways is the nervous system consists of neuron and glia cells. So do not forget those glia cells. They are super useful as well as neurons. It's not just neurons up there. Neurons are sensitive, bless them. Slight imbalances can cause dysfunctions. It honestly doesn't take much. Not holding hands enough, too many chemicals, not enough chemicals, not fast enough, too long, too weak, too strong. They're delicate. So it doesn't take much in order to equal dysfunction or maybe a neurodevelopmental disorder down the line. We have little ability to generate new neurons. And this goes back to what you said, Brittany, something called neurogenesis. When we create new neurons, while a lot of scientists believe it is possible, it's not like it happens often. It's not like our skin cells, which are continuously regenerating. And lastly, vulnerabilities of certain neurons may lead to neurodevelopmental disorders. I know we've essentially spent a whole hour talking about a whole journey just to get to this end sentence. But if you want to know why do we have neurodevelopmental disorders, it's mostly likely to everything we spoke about today, and only just one of those things going wrong or multiple going wrong that can equal how we are. Now, as we know, every single person is different, and because the number of factors and variations on how our brain talks to each other is nearly endless, so it makes sense that we're all different, and little labels that we use day to day, like dyslexic, dyspraxic, whatever, are very limiting and not completely helpful for everyone. Now, I'm just going to round up what I always have to do. If any of you are neurodivergent, give us a call. Especially if you're in the UK, we can get you support. Typically, we can get funding. Not to do any brain scans or anything fancy, but maybe some special equipment. The next session, if you enjoyed this one, is the science of schizophrenia. Ooh, and you think, how does schizophrenia relate? Well, how we talked about how when neurons are stressed, they die or they don't function correctly. A big part of psychosis or schizophrenia is due to stress and due to the connections not working. So essentially, if your neurons are really struggling to communicate and they are stressed, they are overworked, your little janitors in your brain are going on like union strikes, that might lead to schizophrenia. And that is what we're going to be talking about next week. So it's a really nice progression and a good follow-on. Go on our Eventbrite and search Science of Schizophrenia, and you'll be able to sign on. We also have webinars that are recorded each week, and this one will be uploaded relatively soon. So do subscribe, click that little bell icon and all that, and you can sign up. Oh, thanks, Brittany. And here's our contact information if you want to get in contact. My name's Nat, N-A-T, so it's nat@exceptionalindividuals.com. If you want to drop me a message personally with any follow-up questions, I will get to see you again next week or in some future week sessions. But thanks, everyone.